marine organism, you cannot base your organism on metal ions which simply precipitate in seawater, clearly. Vice versa, the metal must also be present in a usable form. For example, zinc is very, very abundant. It's almost everywhere, but a lot of the zinc is actually in the form of zinc sulfide. Now, you all know from your inorganic or analytical chemistry lab courses, zinc sulfide is actually pretty insoluble. So, although there is an abundance of zinc in the form of zinc sulfide, it's not easily used by you or by me or by any human being or by any organism at all, because it's simply too insoluble. And then there are cases, even if something is present in abundance, there may be competitors that will actually limit the uptake. For example, if you look in the ocean, molybdenum is actually the most abundant metal in the ocean, which is quite surprising. And it's there in the form of molybdate, as you would expect. But molybdate, again, is very similar to phosphate, for example. And phosphate, of course, is also present there, and sulfate also in larger amounts. So you have actually three very similar ions, as you can see here. And that will actually, there will be competing. So the uptake mechanism for molybdate will be actually competing with phosphate and sulfite, which are much more abundant. So again, you have a problem. Although molybdenum is available, you need to have pretty specific uptake mechanism that will select molybdate over those more abundant and competing anions. You can think about many details of this, and it all comes down again to, very basic inorganic chemistry very often. And many of the phenomena that you see, for example, which metal is used by which organism, really has to do with relatively simple inorganic chemical questions, and things like availability, solubility, and so on and so on. So a lot of the things that we discover in nature is not magic, but it's really based on very simple first-year textbook kind of chemistry. All right, okay. And then, of course, when we talk about, uptake, absorption into organism, we'll talk about metal-specific transporter and pumps. We have already seen a little bit of this. We'll see a bit more of it. And then inside the cell, we also have to think about delivery to the correct localization of any metal. For example, if something is needed in the nucleus, think of cisplatin going to DNA, yeah? Then, of course, we have to think of a proper delivery system that really also delivers the compound into the nucleus. You'll see questions like this, or, examples for this in my lecture at the end of today. Here's this table that is available in many textbooks. This is just a selection, and we'll talk, first of all, we talk about metal element deficiency, and I was going to point out iron. As you can see, iron, if it's deficient, you suffer from anemia, meaning, sort of your red blood cell count goes down, immune system goes down, and copper, again, has to do with liver malfunction and some secondary anemia and so on. So these are relatively grave diseases. You see that you actually have to take in milligram amounts of these metals here. There are others where you have to have much lower. Vitamin B12 we talked about, pernicious anemia, you know about this already. So we'll talk about the more abundant metals. So in that sense, it seems easier. Yeah? There is more of it around. It should be actually easier. But in some cases, it's actually not. And we'll see also what goes wrong, genetic diseases that go wrong. Here is the things that I will talk about. Iron deficiency, anemia, and copper deficiency is related to something that is called Menke's disease, so we will talk about those. And then we'll talk about the respective metal when you have too much of it. Not a deficiency, but quite the opposite. And these are have also names. It's called either Wilson's disease in the case of copper, or hemochromatosis, in fact, in the case of iron overload. And you can already see when you have these diseases, you can either supplement the missing element or you can remove it. And then, again, questions of chemistry come in. In what form do you supplement the missing element? In what form can you remove something that is actually in your body? I'll start by talking about copper. And this is a picture, it's either this one or a similar picture that you have seen. And these pictures relate to mechanisms of uptake of copper into individual cells. And note the year here. This is taken from a review article from the year 2002 from Amy Rosenzweig's group. And this was about the knowledge that we had back then. So they were already quite happy to see they have identified a copper transporter, CTR1. And you remember this one was also related to uptake of cisplatin that hijacks this transporter. So this has been discovered, and then there are a few, 1, 2, well, there's a question mark, no, 3 maybe. So there are a few more enzymes here that might be involved in copper transport. This picture is important because copper was the first element for which such transporters and so-called chaperones, which deliver the metal from one enzyme to another, they were actually discovered for the first time for the case of copper. So this is an important picture here from an early review in the field, in the sense that it actually highlights the fact that such systems exist. Yeah? It's not a coincidence that copper is, for example, contained in this enzyme, superoxide dismutase. Yeah? Superoxide dismutase contains copper and zinc, and previously people thought, oh, there is this enzyme, and it's built from amino acids, and then somehow magically the metals appear, zinc appears, and copper appears, and they jump into this enzyme, and they make the active form. And if you look at the enzyme and the active site, you will immediately see, well, why should a metal do this? And why should it be a zinc and a copper and not two zinc or two copper or, iron or whatever? And it's not a coincidence, but there is a machinery that delivers the metal from the cell wall where it's picked up and very specifically delivers it to SOD. You can see there are some sort of intermediates here, and people didn't really know what this is. And there is a more recent picture, well, relatively more recent. This is from 2011. I'm sure there are more ones. And what you can see that this picture becomes a lot more complicated. So what used to be just 1, 2, 3 or so abbreviations, now there's a whole zoo. There is the copper transporter still, but there are some others here, 1, 2, 3 at least, additional ones. And then there's a whole lot of ATX1, COX-17, CCS1, a whole lot of more other ones. Here you see a whole heaps of enzymes in the mitochondrial context. There are some in the nuclear transport. There are some in vacuolar transport. There is something that has a question mark that is known to be there but hasn't been identified yet. There's a whole lot more things here that have been discovered in only about 10 years or so. So this is a very active field. People were concentrating very hard on and copper delivery inside the cells, not least because it's also relevant. I won't show details all of these compounds here. I just want to show maybe 2 or 3 in some detail. What I mean to tell you by this is, as I said, it's not a coincidence. Copper is taking up in a regulated way. Yeah? The copper transporters can be up or down regulated, so you can have either more or less copper. And then it's not free, as indicated here, but copper is always bound to enzymes, and it's handed over from one enzyme or one protein to the next. And then this protein takes it and delivers it again to the next. And then this one goes on and so on. Meaning by the end of the day that you are really, it's like, stepping down the stairs. It comes in here, and then here is the target, and then it's sort of handed down the stairs, one by different enzymes. And all of these processes can be regulated. And for example, if something goes wrong here, then the copper is caught, and maybe it's delivered somewhere else to another enzyme. Yeah? So all these things are regulated, and it's important to realize that copper is never diffusing around freely in a cell as was previously thought, but it's really all the time escorted, if you like, by these so-called chaperones, which are responsible for metal ion transport. Okay, let's look at the structure, and these have been elucidated, affinity, copper binding structures in really great detail. I'm not going to show you all of it. I'm just show you something where this copper chaperone, CCS1 or CCS, delivers copper to superoxide dismutase. You remember, superoxide dismutase is the one here on the left. This is the binding site here. Zinc is already in there. And then here is the binding site for copper. And you can see there is no copper in this particular case. That's because one of the binding residues had to be mutated to stabilize this for crystallization. But in principle, the copper will be delivered here. It will be bound to those two cysteines. There'll be another histidine that comes in. So this makes up the binding site. And what you can see is that the CCS protein binds tightly to the SOD, and then here is the binding site somewhere for the copper, for the CCS, and this is then handed over. So this is a site that has a higher affinity than this one. So when the copper is here and this site is nearby, then the copper jumps over to the site which has a higher affinity for copper. So this is, again, not magic. It's all about binding constants, equilibria constants, and you all know this very well when you do you know, your analytical experiments, you precipitate a metal ion and then you dissolve it again. For example, you precipitate, let's say, silver with chloride, and then you add ammonia and it dissolves again, yeah. So this is all about the same thing here. It has to do with affinity, pKa values, you know, sort of stability constants kind of thing. Here's another example. This is the so-called ATX. I showed you this is an important copper transport chaperone here. And this has again been studied. There is this cysteine XX, so those are 2 amino acids, and another cysteine here. You see this motif, cysteine 15, amino acid 16, 17, cysteine again is amino acid 18. So this is the copper binding site up here, and this has been identified because it was co-crystallized with mercury. Now, all of you know mercury is not copper. It has some slightly different binding affinities. It prefers a different geometry. You can see it's linear, as you would expect here, not maybe tetrahedral or pentacoordinate as copper would like to be. But lo and behold, this is the copper binding site and has been identified here. You can make, if you know this, if you know the amino acid sequence, you can make, you know, model peptides. Here's one. Here's a model peptide. You can study the binding. You can do, you know, any kinds of studies, affinity, and so on and so on. As you would for a model compound. Here is one that is really interesting. I told you it has to do with affinity constant. So whenever you have a delivery from one protein to the next, what is going to happen? Of course, the copper will jump from the protein that has a lower binding affinity to one that has higher binding affinity. Yeah? So when you start out with a copper transporter, you have to have a decent affinity, so it picks up copper, but it can't be the tightest binder, because otherwise the copper would be trapped here and it would never get out of the transporter again. So the next molecule down the line must have a slightly better affinity than the copper transporter, and the next one even better, and that has been confirmed. Yeah? You can measure stability constants very easily, and exactly this order of events has been confirmed. And here's an interesting trick that nature plays. This is one of the copper efflux proteins, so-called Cas-F protein. It has been crystallized. Here is actually a copper ion present. You can see, again, I think there is a methionine here and a histidine. So it's 2 sulfur methionines, 1 histidine, and probably some water molecules. And see what else is up here. This is a tyrosine, a tryptophan residue. And the tryptophan, this part here, sort of this double bond here, is actually relatively close, in the order of 2.5 angstroms from the copper. So this is almost like a pi coordination of this double bond to the copper. You could talk about it's almost like an organometallic interaction. And this is well known in organometallic chemistry. Ethylene complexes of copper are relatively stable. I mean, I know this is not ethylene, it's an aromatic ring, so, you know, I mean, okay, fine. But in principle, pi bonded organic molecules can bind especially to copper 1. If this is reduced to copper 1, it actually has quite a high affinity for these. It come on and it can come off very easily again. So you can easily imagine that this is a really good way of labelizing the copper. Yeah, you can imagine it's kind of stabilized while it's in there. Then there is a protein-protein interaction. The tryptophan is removed, and then suddenly you have an open coordination site. Of course, affinity is much lower, and then the copper jumps out onto the next protein. So you can easily imagine what kind of tricks nature can play with these, you know, using such motifs, let's say, from organometallic chemistry, by loosening up coordination sites and then destabilizing the copper such that it wants to jump out to the next protein. And this is what people believe is exactly happening here. Personally, I like the structure very much when it came out. I think I wrote actually a commentary on it when it came out, because it's basically an organometallic. It can be viewed as if it's an organometallic copper complex in nature, which is quite unusual and nobody had seen at the time. This was discovered by Tom O'Halloran's group. Tom O'Halloran and Amy Rosenzweig are the main 2 players in these copper transport stuff, and it was discovered, as you can see, in like 10 years ago. Okay, you can determine these affinity constants and so on. And as I said, Tom O'Halloran is like a leading player in that field. And this is a very important paper in Science, where Tom, by using the known chaperones and the known copper binding enzymes, I mean, there's just a small handful. This is 15 years ago. This is when the field started, yeah? And at that time, he was using the affinity constants, which you can determine. You can determine binding affinity for any, you know, metal by simply sort of this kind of titration experiments with the enzymes that you had or proteins that you had identified. And by doing these experiments and doing some simple calculations, calculating the amount of copper that is there in a cell and how big is the cell, so that means the concentration of copper is like this, blah, 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 blah. You can work all this out. And this is done in a science paper. And doing all this, Tom worked out, and I think it's somewhere on the slide here. It's not? Oh, maybe not. Okay. Tom worked out that there is rarely a single free copper ion, but the affinity of all the proteins is so high that it mops up any free copper ion that would be around. So on any and all of the copper ions will be actually bound to proteins. And one way of seeing this is so-called copper ion sensing. This is the so-called QR sensor protein, which actually recognizes and binds copper. And if there would be too much copper, then the affinity would get out of order. You would actually have free copper, and then this QR sensor protein would actually bind the free copper, feel that the concentration is so high, and then it can downregulate, for example, the copper uptake receptor or other things. Yeah? So there is a well-matched affinity. It's a balance of affinities, yeah? And one here that actually senses it is sort of right in the middle. If copper concentration gets too high, then the affinity of this protein is essential in order to bind this copper and send some signal to the cell. We have too much copper, we have to react, we have to do something. And it does that by binding to the DNA and regulating the transcription sites and so on, okay? And you can imagine this then goes to the DNA and the protein regulation. Okay. So this is what can happen. So now you have at least a view without, really seeing all the details, but you have a view that there are proteins which sense the presence of copper. And in this way, we can see whether we have enough of it or not yet enough. Too much of it or not yet enough. And there is this one here that I want to talk about. This is so-called Menke's disease. It's a deficiency in copper. So if all the machinery works perfectly well, and even with the QR receptor here sensing, we have no copper in the cell. We need to do more. It actually binds to DNA. It upregulates all the other enzymes, but still, there simply isn't enough copper in it. So this is what we call a deficiency in copper inside the cells. And this happens to some people. It's not very common. It's a relatively rare disease, but it has to do with a genetic defect on the X chromosomes. And this gene defect, as we know today, is linked to a copper-transporting ATPase. It's called ATP7A. And this has been isolated. The gene, can be transcribed. You can see what does it. You can search for the protein. It was identified. It was crystallized, and so on and so on. So now we know really quite a lot about this protein here, ATP7A, ATPase7A. It's, as I said, intracellular copper transport. Something goes wrong here. Neurodegeneration, so it's like you have a sort of like, you can't really move your arms properly, for example. And if it's untreated, then children already who have this disease even die before the age of 3. So it's a fatal disease. It's really, really bad. But there is a very simple cause for it. And treatment, as you can imagine, you can try and treat it by simply giving these children more copper. Yeah? Now, eating copper chloride is maybe not the most pleasant thing, and also it doesn't quite work. Simple copper salts are not really effective, mainly because they are not taken up easily. Yeah? If you eat a spoonful of copper chloride, it goes through your stomach, your intestines, and it leaves your body again almost untouched. Yeah? So the bioavailability of copper in the form of copper salts is low. You need to do something better. What is typically done is that copper salts are mixed with histidine. So it's either the amino acid or some preparation thereof, and it's something that is typically described as copper bis-histidine complexes. I think actually this is, again, a whole mixture of things. Yeah, it's not really well characterized chemically. There's certainly several species present. If you just mix copper and histidine, you get more than one single compound. But in any case, in this form, it is actually taken up under physiological conditions. So you can actually have this. And it just leads to more copper being presented to the whole system. And then, you sort of increase copper concentration massively on the one side. And then a little bit of this trickles then finally down into the cells. Yeah? Okay, so this, since has been known, it can be treated. It is not fatal anymore. it is recognized, and you actually have a test now, because you can take the blood from the children, and you can easily test for this genetic defect. And if it's positively tested, then you know what to do. The person has to take more or less copper preparations from the pharmacy. This actually you have to take more or less your whole life, but it's not really harmful. So, if it's well regulated, you can check your copper levels in the blood, and if that is finally okay, then you'll be fine, and you can live actually very long. The other case is something that you can see here. This is copper overload. That's the opposite of it. That is also a genetic disease. And it was first described by a guy called Samuel Wilson, and that's why it's called Wilson's disease. Yeah? This was about 100 years ago, a British neurologist. Again, as you can guess, when it's a neurologist, it leads to neurologic disorders. Again. And it was realized by this person that it probably has to do with too much copper in your body. Yeah? In 1948, this was realized. And then very, very quickly, if you ask a chemist and say, well, I have too much copper, how do I get rid of it? You would say, well, you apply a chelator, a suitable chelator. The chelator will chelate the copper, and then if it's the right one, it can be excreted. And this is exactly what happens. And you use penicillamine as a very simple molecule. This is non-toxic. It's actually a natural molecule. It's non-toxic. You can take it. It will chelate your copper, and it is then metabolized and excreted from the body. Like in Menke's disease that we just discussed previously, here is also a genetic background involved, and this was discovered about 20, 30 years ago, is an autosomal recessive hereditary disease. So you get it from your parents. This is what hereditary means. And autosomal recessive means that if you have, you have two copies of the gene, and one of each is sort of affected, and then if you combine these, men and women, you can either have a chance where you have two healthy genes, and then you have a person that doesn't have the disease anymore, doesn't even carry the genes, or you can combine them in such a way that the two defected genes are combined, and then this is an affected person, and this person will get ill, or you can have any combination in between, and then you have the same as before. Those people carry the defect, the gene defect, but it's not obvious. They don't deliver the disease. So this is what means autosomal recessive. You will see another case in a second where just one copy of the defect gene is enough so that the disease actually develops. In this case, the disease only develops when from mother and father the two defect genes come together. And of course you can see if you have one person that has one gene defect and the other is okay, and you have a healthy person, then you will never get the gene defect. Okay? Because there will always be one healthy copy, and that's okay. That's enough. It's a relatively rare disease. 1 in 30,000 people get it, so that's not really a great deal. We know what is wrong. It's on chromosome 13, and again, the gene codes for an ATPase. And this is associated with copper transport into the bile and copper excretion. So in this case, copper excretion is hampered. It's destroyed. Something is wrong with getting rid of copper. What happens? If the exit is blocked, then copper will accumulate inside the cell, and more and more and more and more, and then suddenly you have too much copper in your cells, and something goes wrong. And in this case, the copper builds up in the liver, so you get liver damage first, and then it's released in the blood, and finally to the brain, and then you get neurological disorders. As you can imagine, if you have too much copper in your brain, you have associated oxidation chemistry, for example, activation of oxygen, and then things get oxidized in the brain, and this leads to neurological disorders. For example, signs of Parkinsonism or dementia, things like this. Okay? As we said, there is now too much copper in the cells. It's a very easy way to cure it, and this is the molecule that you take. This is penicillamine, and it was known previously, and you might actually have used it in one of your experiments. We do an undergraduate lab where we actually chelate copper to the simple molecule penicillamine. And you can determine the binding constant, for example. You can determine the stoichiometry very nicely. And this is a very good chelator for copper, and it has this medicinal background. It's actually the D-penicillamine that we need. You can actually help by simply adding more zinc, because zinc competes with copper, but it's not harmful. So you can help by adding a little bit more zinc and then a metal chelator. And typically, you have to take 1 gram per day, which is quite a lot. Luckily, this thing doesn't really taste very much, so simply take your pills and, you don't really feel it. But of course, you can imagine if you have this gene defect, the gene defect cannot be easily corrected, so basically you have to take this compound all your life. Yeah? If it's diagnosed early, and if nothing changes, then even at age 60 or 80, if you don't take it, copper will build up and you will develop symptoms. So you have to take the compound more or less all your life. The good news is, as I said, this doesn't taste bad and it has very little side effects. There are some long-term effects here, but I think this is relatively minor. So it's actually a good way of curing people. And, you know this from your chemistry lab. It's simple chelation, agent here. You can imagine how this binds to copper. There's a couple of binding sites. It's a sulfur here, amino group. So it'll probably do some chelates and then bind the copper and is excreted. Okay, so this was the case of copper. The other metal that I would like to touch is iron. That's also redox-active. Iron, as we saw on Monday, is the most abundant metal. You have about 5 grams of iron in your body. As I told you on Monday, if it's twice that much, if it's 10 grams or so, it's already too much. You're already ill. You can see the symptoms. I'll actually show you some of the symptoms here. And if it's not enough, say if it's less than 3.5 grams or so, then you also fall ill. You get tired and you sort of you don't feel well anymore. You may faint or so. Especially women know this very well. So you also know what to do. You can take iron tablets. You can buy from the pharmacy. You can buy iron either as iron chloride or some more fancy ways of using iron. So this is well known. You know what iron does in your body. It works in electron transfer proteins. It's involved in small molecule activation, all kinds of metabolism, oxidative damage. So iron is really, really, really important. And the key thing to remember here, and nature is very clever in using this, is that in biology, basically we have 2 forms of iron. That's what we call ferrous iron, which is iron 2+. It's a D6 metal. And then there is ferric iron, which is iron 3+. And that, of course, is D5, and it has 17 electrons. And you can see the different forms here. This is the cyanoferrite ions. And if it's iron 2+, it's almost colorless. It's slightly yellowish. And then if it's the iron 3+ in hexacyanoferrate, then it's this dark red compound here. So those are the 2 forms in which iron is important in your body, iron 2+, iron 3+. And we will see in a second how nature plays with these 2 forms very, very cleverly, and it relates to the properties. This is a number of iron proteins. You probably have learned about this in your bioinorganic classes. But the one thing I want to point out is that most of the iron, we have maybe something like 50 milligrams iron per kilogram body weight, maybe a bit more. And half of it, more than half of it, is actually stored in hemoglobin. So this is the iron, the oxygen transport molecule in your body, in your blood, and you can see most of the iron goes into this. And then there's a storage form of iron, which I will talk about, ferritin and the like, and this has about, like a fourth or fifth or so, right? 20 to 25%, and in the case of female, it's typically less because typically women are losing more iron naturally. So this storage is always a little bit depleted, and at least when you're young and when you're still having children and so on. Okay, here's what happens when you have either not enough or too much iron, and you can already devise this from what we just have said. The main thing, if there is not enough iron, then clearly you're not getting enough iron in your hemoglobin. This is the most affected immediately. And if you don't have enough oxygen transport, what do? It's called anemia, and you feel tired, and you're breathless because you don't have enough oxygen. So all these things here, you can directly devise from the numbers that you have just seen. Also, this is a bit worse. Your immune system gets affected, so you get infections easier, and there are some other disorders here also in your heart, which isn't so bad, which isn't so nice. And then there is overload. Of course, you will see actually the skin coloring, and you develop maybe diabetes and so on and so on. Okay. we'll talk about, we have to think about some general considerations because this is quite important. There are the two forms here, iron 2 plus and iron 3 plus. And if we talk about solubility, yeah, we see here, for example, the hydroxide is much less soluble than the iron 2 hydroxide, which leads to the fact that in water, for example, seawater, when you have iron in seawater, you have a very little, at pH 7, a very little concentration of free iron, really minimal. Whereas if you if you reduce it to iron 2 plus, you have a concentration of almost 80 millimolar. So this is a high concentration. This would be a lot of iron present. And iron 3 plus is basically precipitating out of solution to a very small amount. This is in fact what happens in our ocean about 2 billion years ago, when the atmosphere of the Earth, which was at first, after the Earth was created, you had a lot of oxygen, hydrogen sulfide, so the atmosphere was reducing. It was all iron 2 plus on the Earth. And then photosynthesis set in, higher organism coming up, photosynthesis started. They started to produce oxygen as a result of photosynthesis, and suddenly the atmosphere changed from being mainly reducing to mainly oxidizing because of what we have now, 20% oxygen in our air. Yeah? So suddenly the atmosphere became oxidizing, and all the iron 2 plus that was dissolved in the oceans and in the water suddenly was oxidized to iron 3 plus, and it all precipitated. So the ocean, if you now look nowadays, you have very little concentration of iron in the ocean because it's all iron 3 plus and it's all precipitated. Yeah? So any organism that lives in the ocean will not have, for example, hemoglobin. Oxygen transport in marine organism is never organized by iron, simply because there isn't enough iron. They can't get the iron. What do? They use copper instead, and they have copper enzymes, copper proteins that do the oxygen transport, because copper is relatively well soluble in comparison in water, and they can easily get it, and it does basically the same kind of chemistry. Copper 1, copper 2, you can have copper binding, reversible, and so on and so on. Okay, so those are things that we need to keep in mind, and of course what is true in the ocean is also true in our body. So iron 3 plus tends to be highly insoluble, and iron 2 plus is kind of, easily mobile, has a higher concentration. So this is exactly what our body is also using. It's using this equilibrium, or it's using this redox chemistry, shall we say, in order to make iron either available or unavailable, depending on what the oxidation state is. I told you that the copper chaperone, uptake into cells, copper delivery to certain proteins, this has really been well researched. Well, a similar picture exists for iron. This is a picture similar to the copper one that you have seen before. You see a whole range, at least 4 iron transporters. Here is 1, 2, 3, 4 are known. There is another one here. The intracellular machinery also exists. And you see here, this is from this 2011 review that I pointed out. And but the whole picture is actually much more recent than the copper work. The copper work started back in the 90s, and this is maybe like 10 years more recent. But we also today, we know really a lot about all the proteins that either take up iron or deliver iron to certain target organisms or organelles. Excuse me. Inside your body and my body, as I said, there is a finely regulated equilibrium of iron species, and basically this is the overall picture. You take your iron with your food. Whether you eat meat or not doesn't matter. Also, a lot of vegetables contain reasonable amounts of iron. You take in about 10 to 20 milligrams per day. If you eat normal, if you eat just diverse food, an apple and some vegetables and maybe some other things, some paneer maybe if you're vegetarian, you will take up enough iron in the order of 10 to 20 milligrams per day. It goes into your stomach. Sorry, this is in German, but I guess you can read it. It goes into your stomach, and then into your blood, into your plasma, you have a tenth of this. So 1 to 2 milligrams per day enter from your stomach into your blood. And this is enough, because you excrete, again, excretion is down here. Look at this, 1 to 2 milligrams per day. So by magic, the intake is about the same as the loss that you have per day. Now, if you lose more, of course, let's say you hurt yourself, you lose a lot of blood, then you're losing quite a lot of iron, then you have to intake more. But in principle, this here is an equilibrium, and it works without much regulation. By the way, this excretion here is not regulated. It just happens. You know, you lose hair, you go to the toilet. In this way, you always lose some iron. You sweat, you always lose some iron, inevitably. It's not regulated, it's just happening automatically. Here you can see those milligrams go mainly into hemoglobin, as we saw, and some other iron enzymes, and so on and so on. So the important thing here to note is this, that iron 3+, which we noted is very insoluble, is what we call the storage form of iron, and iron 2+ is what we call the transport form. So in your plasma, iron is bound to transferrin and ferritin, and then it is iron 3+, because it is stored. And if we need it, it's reduced. This is the storage here. Hemosiderin, ferritin is your iron storage proteins. If we need it from here, it's reduced. It gets iron 2+, and it's delivered to hemoglobin or myoglobin or other iron enzymes. Yeah? Okay. Here's a slightly fancier picture about iron absorption across the intestinal tract, but it's again the same thing. You know, this is your dietary iron. Let's say it's in your food. It's iron 3+, is insoluble. It needs to be reduced to iron 2+, and as iron 2+, it actually passes the uptake mechanism, and then here it may be stored again as iron 3+, yeah, and then if it's needed, it is iron 2+, and it's passed on to further transport. So again, you can see here insoluble form dietary iron mobilized to iron 2+, mobilized through, for example, cell membrane into proteins, and so on and so on. So this is the key here, and you already, you can understand this from very simple basic principles. This is a molecular view of how iron is stored. This is ferritin, a so-called iron storage protein. Ferritin is a fantastic molecule. I think it's totally fascinating just to look at it. It's made up of 24 identical subunits. Those subunits are those helical, 4 helix kind of bundles, and out of those 4 helix bundles, 24 of those come together, and they form something that looks pretty much like a football, like a soccer ball. So it's a round thing. It has this shell made up of these proteins, but is hollow inside. So there is a void sphere inside. And into this hollow inside of the ferritin, you can fit in about 4,500 iron atoms. And they're not there as iron, of course, but they are, as we said, storage form. So it's going to be iron-free. It's going to be insoluble. What is it going to be? Well, it's going to be like rust, basically. Yeah? It's iron oxide, iron hydroxide. It's actually a mineral very similar to a mineral called goethite. And this is a precipitate of iron oxide, hydroxide inside this ferritin cage. And how do you get the iron out again if you need it? Yeah, say you lost blood, you want to get your iron back from your storage form. Well, you have to inject electrons, and then this is mobilized. You get iron 2+. This is more mobile. It's more soluble. It leaves the cage and is delivered to wherever the iron is needed, for example, making more hemoglobin. So this is a pretty clever view of it. This is actually from a model for the iron oxide core in ferritin, and here you can see some binding to the outer shell, but I think we can just move on. And this is the genetic or the DNA view of it. So if we have iron, it's actually sensed by an intracellular iron sensing protein. This is called the iron responsive protein, the IRP. There are several subtypes, but just assume it's just one. And this iron-responsive protein binds iron, much like the copper sensor, QR, that you have seen previously. So this is a similar kind of thing. It's a protein which has a certain affinity for iron. And if there's not enough of it, then the iron sensor is not loaded, and it binds here to the messenger RNA of transferrin receptor and ferritin. So it says, we need to have more transferrin receptors, so we need to get more iron into the cell, and the ferritin, the storage, is downregulated because we need to mobilize the iron from our ferritin. We don't want to store it, we want to mobilize it, so you reduce the number of ferritins. On the other hand, if there is enough iron, it binds to the iron-responsive protein. So again, you can see this is just the right affinity. Yeah? If there isn't enough of it, it doesn't bind. If the iron content goes up, then the binding affinity is such that actually it binds to the iron-responsive protein. And then the iron-responsive protein binds to a differently. It forms iron-sulfur clusters, so it's not binding to RNA. So the synthesis of the transferrin receptor goes down, and the synthesis of ferritin goes up, which means synthesis of transferrin receptor going down means we need less iron inside the cell, downregulate the transfer system, and ferritin goes actually up, which means we have too much iron already. Let's store some of it in the ferritin. So you can see this is a super clever system. Yeah? Either this thing here binds to the mRNA and things are regulated, or it goes into something else, is taken away to make iron-sulfur clusters, so there's no binding to mRNA, and then the system automatically goes the other way. What you can see here, that this is all about the iron uptake. Yeah? Only the uptake and the storage is regulated. And as I told you, excretion is not regulated. Your body and my body assumes that we will always lose iron, no matter what. Yeah? And this happens. You lose 1 to 2 milligrams of iron per day if you're healthy. As I said, if you're bleeding, you lose more of it. And then this mechanism here sets in. So the IRP senses not enough iron, and those things here happen on the left-hand side. And this is very important to realize. As we saw in the case of the QR, it's up and down regulated. So it's excretion as well as uptake. In the case of iron, only the uptake is regulated because the iron is actually the excretion is adjusted and automatic, and it's always going to be there. It's always going to be the same amount if you're healthy. And of course, under normal condition, all these things here are in equilibrium with your excretion, so there's actually not really much regulation needed. Let's look at what happens when things go wrong. For example, when you have too much iron, so when the uptake mechanism is out of order, then you develop what is called hemochromatosis. And as you can see, this has to do somehow with hemoglobin, hemo something, and chromatosis means color, and this is what happens. This is a normal hand, and this is a hand of somebody who has this disease, hemochromatosis. You can see this is not a natural color like you are colored, but this is kind of rust under the skin. Yeah, you can see it's sort of brown, reddish color. It's much like iron oxide. So basically that person has really, under their skin, has really little bits of iron oxide, or in their skin there's little bits of iron oxide. And that's the color that you are seeing here. So this is, again, a genetic disease. We'll talk about this in a second. It was discovered already in the 1860s and by a guy called Wilson, and that's why it's called Wilson's disease. And it has to do with the iron loading. It has to do with the transferrin receptor being always saturated. So the transferrin receptor can't really be downregulated. It just takes in too much iron, all the time. This regulation mechanism is out of order. Transferrin just goes crazy and takes in all the iron it can. This is a disease which is, in this case, it's called autosomal but dominant, which means if there is a mother or a father, and let's say one of them has the affected gene, as soon as this copy here is anywhere, then it will automatically, the children will automatically develop this. So other than, in the case, in the recessive diseases where we had just one healthy copy is enough, here the other case is the other way around. Just one affected copy is enough, so the person actually develops the disease. So only if you get two healthy genes together, then you will have a healthy child. Otherwise, it will have this hemochromatosis. Okay, you can see here there are some numbers here, and you can also see what is actually going wrong. Some genetic diseases here, and basically gene controls amount of iron absorbed from blood, and this is dysregulated. There is no reduction of iron uptake, even when all the ferritin stores are full. This is what happens, and you can understand this, and there are some symptoms that you actually see. Again, question is very easy. What do? You have too much iron in your body. Well, much like copper, chelators is the answer. And here's what people do. Chelation therapy. This is a compound that has been in use for more than 60 years. Desferral. It's this sort of oligomeric compound here with a lot of carbonyl groups, OH groups, some amino groups. This basically chelates, wraps around iron and chelates the iron and then excretes the iron, picks up the iron in your blood and helps to do more excretion. So this balance of intake and uptake and excretion is actually shifted to more excretion, so to get a new equilibrium with more uptake and more excretion. Okay. There is also another long-term treatment that used to be quite effective, and that is simply venesection. So you simply give blood. You donate blood. Yeah? Every, I don't know, it was initially, I think it's like every week or something, until your ferritin numbers go down, and then it's enough to do this like every couple of months. So it's basically an artificial way of removing blood, and in this way removing quite a large amount. In like a liter of blood actually contains quite a lot of iron, as you can imagine from the hemoglobin numbers. Yeah, you have 7 liters, so it's like 15% of your iron that's in the blood is removed. So that's actually quite an efficient way as well. Or you take this chelation therapy, and both ways it works quite well. Okay. Some general thoughts to wrap this chapter up. We have seen 2 specific chelators. One was the penicillamine, and then this desferal for iron. There are other ones for iron available as well. But some general requirements, of course, the chelator must be non-toxic. You don't want to kill the patient, obviously. It must be a metal complex that is as a ligand and as a metal complex non-toxic. It should not be degraded too fast, but of course it must be excreted by the end of the day. It must be specific, and it must have a high complex formation constant for the particular metal ion that you are looking at. Here's an example that is taken from strontium and calcium. Strontium sometimes occurs when there is a nuclear accident and nuclear power plant going wild. And for example, like the Chernobyl disaster in Europe in the 1980s, 30 years ago. Suddenly a lot of strontium came into the atmosphere, and people were afraid and say, like, can we take a metal chelator to chelate specifically strontium? Well, strontium is just one under calcium, and calcium is absolutely essential for your bones, and especially for children. Yeah? So in that case, if you want to treat it, you have to be very sure that you have something that is highly specific for strontium and not for the element that is just above in the periodic table. Otherwise, you will have some very bad side effects, and you don't want that. So there was a big, there's a lot of, if you look back in the literature in those times, there was suddenly some activity, sudden burst of activity in strontium chelation. Who would care normally about a strontium chelator? But in those cases, people were really, there was a lot of money available for this kind of research very quickly. Okay, here's some examples. This list is by far not exhaustive here. There are for certain elements, mercury, arsenic, copper, lead poisoning. Lead poisoning can be a problem. Nickel poisoning. So there's a couple of structures here, molecules that have been used as chelators. All the ones that are here are actually in the clinic, and they are in the pharmacy. So potentially, if somebody has, for example, let's say lead poisoning, there is an approved chelator for lead that people can take, and that is safe to use and safe to take, and will hopefully help do the job and remove excessive lead, in this case, from your body. We will talk about poisoning. Talking about poisoning, this will be the topic of the next 2 lectures, and it will be those 2 elements here, mercury and arsenic, that I'll be talking about in terms of, really being toxic metals and what actually goes wrong, and what you can learn about, again, what you can learn about chemistry from the basic toxicology of these metals. As always, some literature at the end. We will only have a short break, I would say, 5 minutes or so maximum, and then continue, so we'll be finished by about 4:30 or so. Okay? Short break, 1 cigarette, and then come back, and we go on, okay? We can have questions, if you like. We can have questions at the very end of the next session. I think they fit together well, okay? toxicology. So we will talk about metals or metal ions or metalloids to some extent, which are not naturally in your body. So they're not like iron or copper, which you need. But we will talk about arsenic and mercury, so things that you should not normally have in your body and that will be harmful. And this is a picture that you have seen before. We had this in an earlier slide. And now we have been talking about, deficiency, and we have been talking about the correct doses and concentration here throughout the week. Yeah, we've been thinking about drugs which operate in this regime here. And now, today, we will talk about some examples of toxicity. So we'll talk about the far end of this graph. And just to remind you, we want to talk about species-specific toxicity. So toxicity wasn't really bad by itself, but we really want to concentrate on things which are now toxic for humans. So we are not talking about any specificity, but things that are really just toxic for you and me, that would potentially kill you and me. And I think this curve we have also seen. If you give a certain amount of a toxic substance, there is a toxic effect, where 50% of the individuals react. This is called the TD, toxic dose, where 50% react. And then sometime later, people die, so it's a lethal dose here. And in case that we have toxic compounds, this lethal dose, of course, is very much to the left-hand side. And this window here may actually be very small in the sense that you have a toxic effect, and that leads very quickly to lethal. So if this curve here moves to the left and there's a very little gap here, then this actually becomes a very dangerous compound or a very dangerous element, let's say. Again, this is this poisoning time course. We have some latency period until we have an onset, and you will see that this latency period here actually becomes very, very important. Here it looks as if it's really a very short time. Within an hour or so, you get the effect of your compound, which is maximal after, what is this? This is 25 hours maybe, so maybe after 3 or 6 or whatever hours, this is the top effect already, and then it goes down. So this would be something like aspirin, for example, where you have a good effect after 20, 30 minutes or so, and then after a day or so, you do not have the aspirin effect anymore in terms of painkiller. But we will see that these things here actually can be very, very important to keep in mind. So there may be a long latency period which you don't really realize. As I said, I will talk about two elements in particular. The first one will be arsenic, and you think back to what we had yesterday, the salvarsan compound. I'll quickly draw on this, and we'll go on to other applications or other cases. And then the second example, as I said, will be mercury, and then we will use that to finish off. So we already had talked about the medicinal use of arsenic compound in this Ehrlich 606 compound, salvarsan, to treat syphilis. I should say that there is a very interesting story that there are people in the Austrian Alps, workers that work high up in the mountain. Maybe imagine like the Himalayan in Nepal or in northern India. People who work high up in the mountain, and they used to eat, basically they used to take some of their soil and grind it up and, sort of dissolve it and drink or eat these preparations. And it turns out that this soil in some parts in the Eastern Austrian Alps, it actually contains relatively high amount of arsenics containing salts. So arsenate, certain minerals that contain arsenic. And so these people were constantly eating low doses of arsenic. And there were so-called arsenic eaters. Yeah? And this is a real story. There's like families where for centuries, when they were working high up in the mountains, where blood pressure, the oxygen pressure is low. So these people were eating arsenic, and it was found, and this is of course what they wanted, that they developed more red blood cells. Yeah? So they had, compared to people who had not done this, they had simply many more red blood cells. And in this way, even when you're high up in the mountains and there isn't so much oxygen available, those people could still work hard, and they were physically stronger because they had more red blood cells and they were taking up more oxygen, very simply. You can think of this when you're an Olympic athlete. Maybe you want to start eating arsenic to develop more red blood cells and be, run faster or jump wider, jump higher or whatever. I don't know. But anyway, this was just a finding. This was known for many, many, many years. And then at some stage, some clever doctor actually thought of this, and today there is a drug on the market which is basically arsenic trioxide, which is kind of frightening because that's also a poison. Yeah? But if you take it in low doses, it is actually used to treat leukemia, or rather it is given to patients after they had gotten chemotherapy in order to help them develop their red blood cells faster. Leukemia basically is a cancer of red blood cells. What you do is you kill all of them. You give them a very high dose chemotherapy that kills the red blood cells, and then you give these people arsenic trioxide so that they make new red blood cells faster. So this is not directly a cure for the disease. It doesn't kill the cancer cells, but it helps the patient recover. And the survival rate when people actually got this are dramatically better than if they don't get it. So this compound here, although it is really toxic and it's arsenic trioxide, I mean, think of it. You can poison your grandmother with it, right? But in low doses, it made it very, very quickly, in record time, to the clinic because the success rates were so astonishing. And, for some people in the Austrian Alps, this was no surprise at all because they had, without knowing it and without having cancer, but they had used the trick that is played here for centuries. Yeah? So I think this is actually quite an interesting story. There is another interesting story here that relates to, that you probably know about. It's about drinking water in Bangladesh. Like in the 70s, after Bangladesh had become independent from India, it was one of the poorest countries in the world, and like people were, dying like fleas, and it was really, really very appalling. Hygienic conditions were really, really terrible in that part of what used to be India. And then the World Health Organization devised a program to drill new holes in the ground and to get drinking water from the ground. So they got wells. Lots of wells were built, actually, and it was later found out that instead of the drinking water from the rivers, which was loaded with bacteria, they got clean underground drinking water. But unfortunately, after a while, it was found out that these people seemed to have what looked like arsenic poisoning. Yeah? And maybe the picture from the people, they had skin colorations, and that really looks like arsenic poisoning. I'll come back to that point here later on because it was then found that there was quite a high amount of arsenic in these wells, and it was estimated that half of Bangladesh or so was affected. So it was really quite bad. And it was intended to do a good thing, provide clean drinking water, but unfortunately in that area of the world, the drinking water had relatively high arsenic amounts. And I'll come back to that in a second. There's some really nasty applications, in quotes, of arsenic-containing compounds as well. For example, it was used in the Vietnam War by the Americans as a herbicide to basically remove the leaves from the trees and things like this. So it's really, really not pleasant at all. Talking about arsenic trioxide, it was actually used as a poison in the medievals, hopefully not in this country, but certainly in Europe. We have all these many, many small kingdoms and kings, and they were constantly fighting each other, and they were fighting the Pope, and the Pope was fighting them. And one means, this is the medieval times, right, 1,000 years ago or so, but one means of fighting the other family was actually poisoning them. And one way of poisoning that was very common was actually by poisoning people with arsenic trioxide. Maybe about Miss Marple, the detective. There is a famous case when she solves a case of arsenic poisoning, one of the criminal stories, there's a real background to this. There was this famous in Italy, this is the Borgia's family, and they were a very big family. They were very influential, but they basically, literally, they extinguished the family by poisoning each other. So this family disappeared almost because they had basically killed each other more or less by arsenic poisoning. This was, I say here, this is common in medieval time. I mean, it's not as if every European was, poisoning their family, but it was a relatively frequently used poison, arsenic trioxide, arsenic pentoxide, up to the 19th century when forensic science started. And you may have done this. We don't do it anymore in our lab courses, but you still may have done it. There is a famous probe for arsenic, which is called Marsh's probe. Are you using it? When you're reducing, you have a sample, you think there might be arsenic in there, you're reducing it by zinc and HCl, so you're making in situ you're making dihydrogen, and that reduces the arsenic, and you get arsine, ASH3 out. You still do that? No. Okay. I mean, we abandoned it also. Like you, we don't do it anymore because it's, arsine is highly toxic. Yeah. You don't want to kill your students by, having them breathe arsine. But it's a very potent probe. It's a very, very sensitive probe. So basically you take, when you believe this person might have been killed by arsenic poisoning, you take some of their stomach, what remains in their stomach, you boil this up in the Marsh probe, and you collect what comes out of it, the arsine, and that forms, if you just hold like sort of a beaker or so on top of it, it forms this sort of silvery, shiny thing when it actually decomposes, and you get arsenic metal basically back. So it's a very sensitive probe. It works on milligrams of arsenic in these intestines. And when this was discovered, that was the end of arsenic poisoning, because then suddenly the police and the judges had a way of proving that that person was poisoned by arsenic. And previously, why is this so common? Why was it such a good, in quotes, way of killing a person? Because the symptoms of arsenic poison are very much like the symptoms of a very severe infection. Yeah? As if you had a stomach infection. So you feel really bad, you're vomiting, you feel really ill, and you have to lie down, you're fatigued all the time, and so on. That is pretty much what it feels like when you have a very bad, maybe some of you have had it, like a very bad, food poisoning or stomach poisoning or stomach ache or so. That is pretty much like arsenic poisoning looks like. So the doctors at the time had very, very little chances of deciding, is this an infection or is it a poisoning? And of course, medieval times, 1,000 years ago, infections were very common. So people were having infections all the time. So there was nothing special about it. And arsenic poison pretty much looks like it. Yeah? And so this only stopped when forensic science developed, and where is it here, Marsh test here. When forensic science developed, and suddenly the scientists could come in and prove that this was not an infection, but it was deliberate poisoning. And then these things stopped because it became much too dangerous for the murderer, because there was a good chance that the judge would actually be able to prove that this person has been poisoned. There is some toxicology here of arsenic compound. Again, it's a question, what do you mean by toxic? For example, arsenic trioxide, arsen in the oxidation state +3, is much more toxic than the arsenic pentoxide. Okay? So the trioxide is really what is used to kill people. Also extremely toxic, I told you, is this arsine, ASH3, whereas trimethyl arsine, so the methylated version, although it's arsenic-free, isn't actually that particularly toxic. But it's degraded here, it's easily oxidized, and so some things happen here. This is what I said. Poisoning looks like a severe infection, whereas the body adapts to subacute doses. And this is what the arsenic eaters would to do. Yeah? They didn't kill themselves by poisoning, but they took sort of smaller doses, which just made their blood develop faster. You shouldn't do it too long, though, because you can have liver failure as a consequence, as a late complication after years and years. You actually might have known this. Some of the kings, it is said that the Egyptian emperor Cleopatra, she actually was said to eat small doses of arsenic with her meals in order to prevent people from poisoning her. So you get used to it. Yeah? So to kill Cleopatra, you would actually need maybe 2 or 3 times the normal dose. Yeah? I don't know whether that is true. I mean, we have no way of knowing, but it's reported from her food, from her diet, that apparently it contained some arsenic all the time, and this was intended to make her immune against arsenic poisoning. There is a famous story, okay, we can go through this. This is all the arsenic chemistry. Talking about arsenic poisoning, there is an interesting story that you may have heard about, and if not, I'm going to tell you about it. This is the fact that in the 19th century, it became very fashionable not to have white painted walls like we have here, but to have wallpapers. And the rich people in the country, in England, for example, they wouldn't want to have white walls, painted walls. They wanted wallpaper on their walls, fancy wallpapers. Very fancy was something that was green, green colors in these wallpapers. And this is called, the pigment that was used at the time was Scheele's green. So this is from a scientist called Scheele. He invented this color, and it was basically a copper arsenide. Okay? So that was what was the green pigment that would color these wallpapers. Now you can imagine you're in it's not medieval anymore, but it's like 17th, 18th century England, 19th century England. Small houses, it's raining all the time, it's damp, it's miserable. I mean, you don't want to live in England in that time, right? But there's a lot of moisture. There may be some heating inside, but it's wet everywhere. It's not well ventilated. Yeah. So what is happening that actually microbes were developing here in these wallpapers, and people became, as you can see, very rich with this. But somebody suggested very quickly in the early 19th century already that some toxic odors might arise from these wallpapers. And it was later found out what it is. It was arsine itself or trimethyl arsine, yeah, that came out of these wallpapers. And what was happening is that you have arsenic oxide, or some, anionic form of arsenic oxide, and this would be methylated by cobalamin. The bacteria on the wallpapers would produce, like bacteria do, like we discussed on Monday, they will produce cobalamin, methylcobalamin, vitamin B12, methylated vitamin B12, and the methyl group can be transferred to the arsenic. And in this way, you or the bacteria would make trimethyl arsine, and of course that is a toxic poison. Yeah? It's either this one here or this one that was actually given off. So the wallpapers would regularly and constantly, especially when it became a bit warmer in the summer and it was still wet. England rains a lot, yeah, always wet. So during the summer, when it is wet, suddenly you would get all these bacteria growing and they would produce trimethyl arsine. And if your house is not well ventilated, I mean, this is not a problem. Yeah, we have all the windows open, all the doors open. But in England, because it's a bit cold, you want to keep your doors shut. And then people were getting poisoned by arsenic compounds. It turns out, this is quite bad for some people, and people were suffering from it, and so on and so on. Some died, and then this wallpaper thing was abandoned. And nowadays, of course, we don't have minerals anymore, but we have sort of organic green colors that are used. So it is supposed to be safer. But there is an interesting story behind it, and this is the story of the French Emperor Napoleon. There has long been allegations. Napoleon was in the early 19th century. He was the emperor, and he ruled almost all Europe. He went way into Russia. He was finally defeated, and then for the first time he was sent to, I think, some island nearby France. He came back. He ruled another 100 days, and then they finally got rid of him, and they sent him to St. Helena. So this is some island way out in the ocean, yeah, far, far, far away from France. And he died there. And of course, he had some followers still back in France. And when he died, his followers said, "Oh, he has been poisoned. He has been poisoned by the current French king who wanted to get rid of his competitor." Yeah? And these rumors never disappeared. Yeah? Throughout the centuries, there were always like people who were saying, "Oh, Napoleon was poisoned," and they came up with all kinds of bizarre arguments, pro and con. Was he poisoned? Was he not poisoned? So finally, towards the end of the 20th century, we're talking about 1980, 1990 now, Napoleon was brought back to France and he was buried in France. People actually took out the body and they took samples of his hair and analyzed for arsenic. And nowadays we have analytical methods that are so good, you can actually analyze on a strip of hair, whether there is arsenic in it or not. And because your hair, I mean, your hair, not mine. Mine falls out quickly, but basically, your hair grows for a certain amount per time. So if you have long hair, like these women here, for example, Anupa, like your long hair, if you analyze your hair, you can probably see whether you had arsenic in the last 2 or 3 years. Yeah? So like the tip of your hair will go back. I don't know how fast your hair grows, but let's assume it's at least a year or so. So you can see whether your husband tried to poison you, yeah, a year ago. You can see that by analyzing your hair. Okay. I'm sorry, I don't think that's going to happen. I know you love each other. I'm just making the example. So, yeah, this is forensic science. And of course, this comes with excellent instrumentation. Yeah, you need to have a highly sensitive detector, and you need to be able to do this localized on small pieces of hair. So this is forensic science in the 20th century, okay, or 21st century, actually. So, highly specialized instrumentation to do that. Anyway, it was found that indeed Napoleon's hair contained pretty significant amount of arsenic. So the people said, finally we have a proof. He was poisoned, The emperor poisoned him by arsenic poisoning. We can see it in the hair. And of course the doubt is, just as I told you, there is the wallpaper story behind it. And the question now is, did Napoleon in his house in St. Helena, which is in the ocean, so it's like wet all the time, and it's further south, so it's warm all the time, did he have green wallpaper with Scheele's green or not? If he had, then this would be a logical explanation for arsenic, and if not, then of course he might have been poisoned. And it turns out that by accident, I think this is in the late 1990s or so really recently, somewhere in Scotland, somebody actually found a painting, some woman that was a caretaker for Napoleon or so, had prepared, for example, paintings and pictures from the house, and it was actually seen that they had wallpapers, and there were even samples of the wallpaper from Napoleon's house on St. Helena were preserved. And so there was original samples from that very wallpaper, and they were analyzed, and of course they contained Scheele's green, and since then there is at least a logical explanation why the arsenic is in Napoleon's hair. That doesn't exclude that he might have been poisoned still, but at least it explains that there was arsenic in his hair, so nothing bad about it. Okay, so I think this is really a fascinating story, and you can see, you can very easily be deceived. If you just look at one facet of the whole story, you can actually be taken the wrong direction. But it also teaches you that you need very good science, and you need to be really a scientist to cover all aspects of the story, and always think one step further and say, is this really the only explanation, or could there be something else? And I think this discovery by this woman in Scotland who found these sort of diaries from the caretaker, that was pure coincidence. That was good luck. Like, she was cleaning probably, some room in her house, and she was about to throw it away when she discovered, this is actually something from, Napoleon's times. All right, fair enough. Wrap up of the story. I'm coming back to Bangladesh. You might have heard this. The Bangladesh story actually, what was happening there is that there was high amounts of arsenic, but the people weren't poisoned by arsenic, but the arsenic reacts with selenium. And what was happening in Bangladesh, that the arsenic-rich drinking water was in reality depleting the selenium in the diet. And because the people as I told you, Bangladesh is like a very poor country. They didn't have a very good and very balanced diet. So typically they didn't get enough selenium to supplement the amounts that they would need. So the arsenic in the drinking water in Bangladesh reacts and forms, for example, this glutathione arsenic selenate anion, for example. That is a species that would isolate it. This can be isolated, and you can see that arsenic sort of traps the selenium and removes the selenium. So what actually happened in Bangladesh is that the people had the same symptoms as if they had selenium deficiency. And that was what it was. It wasn't arsenic poisoning. It was, as a secondary consequence, selenium deficiency. And then you know how to cure it. They can still have the good drinking water. They just need to supplement selenium. And that's what the World Health Organization did in some areas. Just handed out selenium tablets, added selenium to the water, and so on. And then those people were helped. So again, good science, careful analytical techniques, careful experimentation will solve the problem. Do we have a few more minutes, maybe until 1:45 or so, and then we finish and have some break and start with the other lectures at 5? Is that okay? Yeah? Okay. Okay, I'll be quick with my last example, the example of mercury. It's another, sort of fascinating story, and unfortunately some of the stories are fairly sad. But it comes to the point again, toxicity. This is the main topic, What is toxic? Well, for example, elemental mercury is by itself not toxic if you take it orally. You could potentially, I don't recommend you do this, do not do the experiment, don't try it, but you could take a spoonful of mercury, swallow it, eat it, and it would come out more or less the same way when you go to the toilet. Mercury is very, very heavy. It goes through your stomach very, very quickly, and you lose it in your backside. That's it. Not dangerous. It's not going to kill you. But don't try it, please. Don't try it on your dog either or so. It's not a good experiment to do. But, from the physics and the chemistry behind it, mercury, elemental mercury, will not dissolve. It will not go into your blood. It is, of course, very dangerous as elemental mercury when it is taken as a vapor. And you know the thing, when you break a mercury thermometer in your lab, you really have to be very careful and mop up all the mercury and remove all the mercury. Don't leave it on the floor.
That is supposed to pick up telomerase. It's called telomerase is the enzyme that works on the DNA. So it needs to be, essentially, telomerase, the enzyme, it needs to be in the DNA in order to work properly. But the protein biosynthesis is never in the DNA, it's somewhere else in the cell. So telomerase is synthesized somewhere else, and then it needs to be transported into the cell because it's useless anywhere else, because its only function is working in the DNA. How does nature do that? Well, nature attaches a small peptide to the telomerase. This peptide is recognized by the cellular machinery that recognize the protein, recognize the peptide sequence, take the peptide sequence inside, and this way carry the telomerase into the nucleus. So these peptides have been discovered. People have worked on it. The Nobel Prize was awarded about 50 or more years ago for the discovery of this machinery. And we are going to use the type of machinery. We will use such peptides in order to direct, in this case not protein, but in order to direct the metal complex to wherever they are inside the cells. So this is what we want to do. We want to make peptide conjugates of the metal complexes which are toxic in this way in their properties. There are various peptides that we can look at. For example, there are peptides that are unspecific for cell uptake, but they are very good in getting things into cells, anything you want. So you get a large amount of whatever you attach to them into the cells, but the disadvantage would be they're unspecific. There is also quite the opposite. You can have a peptide which binds to a certain receptor very, very tiny, very controlled way, and then the receptor transports the peptide into the cell. This is the intracellular machinery I'm talking about. This is very specific for this particular receptor. Imagine this is a receptor which is overexpressed on tumor cells. And you would have a way of controlling uptake into the tumor cells. Think about the estrogen receptor that we're talking about. That estrogen receptor was needed specifically on tumor cells because it's associated. So we have an example here how specific this can be. Of course, the efficiency may be low because, for example, a tumor cell maybe has only 100 or 1,000 of these receptors. So for a time, you can only get, say 1,000 molecules inside the cell, which is not a lot. And then finally, in the last couple of years, so there's advantages and disadvantages. And at the end of the talk, I will tell you a story about peptides which combine the very efficient uptake and the specificity. So it's like the ultimate system here, if you like. So they combine the good points of the two. They don't seem to have any negative side effects at least. So I will tell you, I will wrap up my talk by talking about these peptides and give you a kind of those previously. But before we do that, as I said, this time we're actually going to go through some of the steps that we have to do. We will talk about peptide synthesis. We discussed earlier this morning with one colleague here, speaking also about peptide synthesis. And this is what we do in our lab. We are not really organic chemists, but we are adopting what organic chemists have developed. And we are using the so-called married-field synthesis, which is a sequential peptide synthesis on a solid support for our peptide synthesis. So what you do here is you take a solid support, so-called acid, you attach the amino acid to it, which is protected, you deprotect the amino acid, and you add the next one. And then you start the whole cycle all over, you deprotect, you attach the next one, you try that, you deprotect, you attach the next one, and so on. Until you have the full sequence assembled one amino acid after another. That's why it's sequential, because we're doing it one after another. Last thing you have to do, you need this one for the resin. You have the peptide, you purify, you are there. You have the final peptide with all the amino acids in the right sequence. nice and clean. The advantage of this strategy is that you do this because it's on the resin, you can add all your reagents in a very big sets. You drive all reactions to completion because you use a large sets of your reagents, and anything that is needed is washed away because the only thing that we want is resin in whatever is attached to the resin. There is something else we need to worry about because we want to introduce metal somewhere in the seed. Now imagine the original Merrifield strategy actually uses very, very strong acid. So it uses concentrated trifluoroacetic acid and it uses cleavage from the resin with neat HF as before. That is pretty bad. I mean, how many metal complexes will survive treatment with strong acid and HF? Not so many. So this is maybe problematic. But luckily nowadays there are milder strategies available. A variety of thickness is possible to use, and we use mild base or cleavage of such technical. So today we have milder strategies which are compatible with the chemistry of many metal complexes. Let's quickly look at the way how we make peptides in our lab. The best established in the old way, actually the way Merrifield do it as well, is simply to do it in a syringe here, which has a thread at the bottom, right? Here's a thread. And what you do is you have the resin in the white powder, right? You have this in the thread, and you can use your solution of your compounds, you pull them up in the syringe, you shake maybe for 15 minutes, you do this 15 minutes, and then you remove the liquid again, you take up another solvent to wash, maybe 2 minutes shaking, remove the washing solvent, and so on and so on. So you can always take up the reagents in the syringe, you shake, and then you remove it like this. Well, and of course the resin, because this is a thread, the resin is retained, or the solvent is reagents or removed. So we have actually prepared this in such a way that we can make the peptides because that metal content is actually the solution tracking. So these are original syringes that students have used in Tract. This is just neurotensin, this is a cobalt complex, this is a ferritin, so you see the iron on it. This is an organic ion, so you can see nicely the different colors. And they are all, because they're directly bound to the resin. The resin is by color. What you see here works very well. As I said, it's a student practical, so we use it for students. And as I say, this is the student methods to make peptides. When you're a bit more advanced in my group, then this is the postdoc and PhD way. It's actually a computer-driven apparatus. There's a laptop next to it. So what you have to do is you feed in here you attach the reagent bottles, various amino acids, you program the computer, you press go, and this may take a whole day, and you shake in 15 minutes. And you can imagine 5 amino acids times 15 minutes, you will do this for 75 minutes. You know what's the egg? Small egg. So this is the student way of making peptides. And here you don't want to do anything, you can bring the copy while the machine is doing the work. I should say, in all honesty, I mean, I have jokes about it, but actually very often also the PhD and the postdocs like the syringe method because you have direct control. You can see the color, you can see what's going on, you can see if there's a composition or something. You can sort of look at it, you can remove the stream, and you can still see in the stream, for example, check that's my reaction on completion, maybe I need to do something here. Here, once this thing started, it will work to the end. If something goes wrong with the tree, or doesn't quite work so well, you have no way of doing it. The computer works to the end. The computer works to the very end. Look at the product. Long. So here you have much more control, and that's why also PhDs, of course, like to use the search method or the combination of the two. And part of this here that can lead to control transferrin research. Without going into details, I will just show you this overview slide. This is the key peptide that we have used for Traficking. This is the sequence of the Traficking. Here you can see the range of different metal complexes that we have attached to the Traficking. This is just a selection from our research. You can see Ferritin here. You can see here's a Ferritin compound. That's one of these regions. Here you have the Chaperone compound. That's why we have used it. Here again is a CPU compound with another ligand. Here are some metal carbonyl compounds. Here's a tungsten compound with an alkyne bond with a gold compound. So you can see that the whole variety of metal complexes, different ligands, attached to this Traficking peptide, which just gives you a flavor of the kind of things that we've done in the last 10 years or so. Just an example. Actually, we have the latest slide probably 4 or 5 years ago. I could put on at least twice as many now. Probably becomes too small to read it, but it's older than what is here. I will go into two specific examples, not in this Traficking, but a lot of peptides. Those other peptides I will use. We will use octreotide, which is a cyclic peptide, as you will see, and this is a very, very good one for receptor-mediated uptake. So this is specific now to the target. Key question that many of you have addressed in the lecture. I have kind of tried to debate it sometimes. We have discussed in various ways. Here is going to be a solution how we can make things hopefully tumor-specific. So let's look at this peptide. Somatostatin is a well-known natural peptide because in your body and my body, it's a regulating peptide and it inhibits the effect of growth hormones. So growth hormone makes the tumor grow, and somatostatin is given to potentially stop the tumor from growing by interaction with these so-called somatostatin receptor SSTRs. These SSTRs are overexpressed in neoplastication, which means, for example, in lung cancer and breast cancer. So here is clearly a way, if we take this peptide here, we can stop the growth of tumors by binding specifically to those tumor-specific receptors. So here's an answer to your question. How do we make these tumor-specific? Well, we pick a receptor that is overexpressed specifically on tumor cells, and then we find something that binds to it. So this would clearly be a good way of doing it. The problem with somatostatin is in this number here. It's the half-life of the peptide in your and my blood. So 3 minutes. 3 minutes is way too short. If you reject, 180 seconds later, half of that peptide is gone. It hasn't even reached the receptor. It's still circulating. It's still trying to find its target. By the time it reaches its target, it's full fraction. So these 3 minutes are clearly way too short. This is because this is a natural peptide, and of course it's a peptide. That's all peptides constantly. There are proteases, but very But luckily in our chemists, the chemists have come up with smart solutions. They have made a shortened and more stable version of this peptide here, and they cut somatostatin to something that is called octreotide. Octreotide, as the name implies, is only what is 19 acids. Okay, so it's 19 acids instead of the whole peptide. It still has these types of the bond here. It has a OH group, and as you can see, it has 2 amino acids. So clearly, this is now much more difficult to degrade, and this is the chemical structure of the compound. Now, yeah, it's not that nice. Okay. So this is also marketed by Novartis. It's somatostatin. It's specific for these receptor subtypes. And here you see the number 3 minutes. That's what happens. 90 minutes. 90 minutes is enough time to target the mind, you take enough, and then anyway removed from the blood so this would be a good target to work on. You can see here the peptide, and we can actually make it. We made it in our lab. We could potentially make it. It is a little bit tricky, but it's not so bad. So we made the resin. We have the peptide synthesis, and you see the side chain protecting groups. finally, here we put on metallosines, for example, kerosene, phenosines, so the metals we can use to put the genetic complexes on this peptide. You can see that we formed the disulfide bond between the two cysteine residues here by adding by removing the cyanide protecting and adding the chelation free. So we have the disulfide bond, we have the metallosine on it, and we added the terminal lysine where we can put on fluorescent in order to see whether we get the uptake into the cells that we want. So this is the final project here, the final peptide, full-length octanotype with the metal complex, with the fluorescent dye. Now we can go ahead and study the uptake. First of all, we were looking at different cell lines, and we were trying to find cell lines which actually overexpress this somatostatin receptor. What we use is so-called real-time PCR, which basically scans for the number of copies of a certain protein on this receptor. So if we look for the messenger RNA of this receptor, and the assumption is that there is a lot of messenger RNA, this will also mean there is a lot of protein. And if there is very little messenger RNA, then of course we expect less protein to be made. We do this by PCR, so by simply amplifying this messenger RNA. And here you can see the more cycles you need in order to duplicate, the less of the messenger RNA was there initially. So here you can see the numbers. So the experiments that we actually do, and here you can see the numbers. For example, you have three different cell lines. The yellow one is the liver cell line, G2 is the liver cell line, or the pancreatic cancer cell. Pancreatic cancer is particularly very pure. So we're hoping to do something. What you can see, the smaller the number, the more of the receptors is, for example, ELA and G2 will be, for example, for the SSTR3 receptor, they're about the same, and this one here, P45 is about 2 orders of magnitude less. Here, the difference for the SSTR5 is actually 5 orders of magnitude between G2 and P45. So we have a cell line that has G2, that has an ELA, that has a lot of the receptor, and we have P45, but it doesn't have that much of the receptor. So does that translate into uptake? Now, if we take our compound and we incubate, for example, the cells which are TR2 positive T2 cells, this is the overview. All these are cells here in cell culture. We added our compound with 5 micromolar, that is below concentration. And we see here quite a number of growing cells. So all the growing cells have taken up the compound. This is acetylated nucleotide, and this is the ferritin derivative. So this is the metal derivative, and this is metal free. If you just compare this, you can see that actually the ferritin is even more taken. So there are some major cells that take up, but also minor ones here in between. All these cells have taken up at least some of it. So that's actually pretty good. This is a sort of normal light microscopy. And so we have a pretty good uptake of 5 micromolar. What happens now if we take the P45 cell line that does not have this receptor, or much less of it? This is the picture you're getting in comparison. This is the P45 cell line. Just compare this for the acetylated or for the ferritin derivative. You can see here again it's more, yes, than this, but compared to this, it's a fraction. It's hardly anything. And look at this as well. Here we're using 50 micromolar, so 10 times more than here, and we're still getting less uptake. So that clearly is an indication that yes, we are using the receptor, and the positive cell lines takes up much more of this compound than the SSTR negative cell. We look further, we can zoom in on one single cell. This is now one single cell. So we really zoomed in massively. And we can see by using this our compound, the ferritin lysine, the fluorescent dye, we can use, for example, some dyes that stain some intracellular organelles, for example, the endosomes or the lysosomes. Here is the lysosomes paradigm, here is the endosome. And when we overlay the two, our compound and the tracker, you can see here it's overlapped really nicely. So green and red together give yellow. So this is a lot of yellow, which means that our peptide and the lysozyme are in the same organelles. Do the same for here, you can already see sort of visibly enough. You can see there is much less overlap. So really we have endosomes, sorry, compound, which is another indication for its mechanism, because it's known that the receptor is actually internalized through the microphage. So exactly as we see here, the compound is internalized through the receptor of sugar. To me, that looked pretty convincing. We have one other experiment to do, and this goes back to structural biology. It has been shown that this peptide oxidation was discovered. It has been shown actually that this high sulfide bond, this is the structure that was earlier produced. Here, this disulfide bond is really, really crucial for activity. So it needs this cyclic form in order to bind to the receptor. So it's found to the receptor. If you open it up, then it can't really fit into the receptor so well. So we can make an experiment. If we're making these compounds experimentally in our lab, we can use the cyclic form, this one, or we can use an open chain analog of the oxidative type. For example, when we just block the fibrates, the cysteines, they are blocked, so they can't form the disulfide, and we expect that this thing here will not bind to the receptor, whereas this one here would. And we've done the experiment, and this is what you get. This is a comparison of the lysine oxidative cyclic one, the open chain. Just compare the two pictures. Here, everything is treated. I would say every single cell is standing in our 25 microgram. And here, same concentration, much less stable. You can see, yes, there is some uptake. It's not zero, it's not all or nothing at all, but it's a lot less. So this is another lesson that we learned when we were working on this. And you also see the bar graphs or the SSTR receptor overexpression. We tend to say, oh, the receptor is overexpressed and it's there and it's not there, but that's not true. It always means it's the more, it's the less. So there are always gradual differences. It's very rarely, zero to 100, but typically somewhere between some more and others less and so on. So you have to deal with this. But I think if you look at these pictures, even if you're not an expert, quantify this a little bit, measure the amount of time, count the amount of those cells and all this. But even if you don't do it, I think you can just see visibly, yes, there is a large uptake. So this is a way of making things really specific. I'll tell you another story about specific uptake. In this case, as I said, we won't talk about into uptake into nucleus, but we will talk about uptake into the mitochondria. And we will use an example of gold compounds. You already know from the lecture on the when we talk about gold compounds, you recognize this compound, Inorganic. Nolte is an anti-alkylic drug, has some anti-tumor properties as well. It's a low compound, very soft mechanism. It binds to redox-active, and by the do that, this redox-active has a selenium atom in the active site in the center. So this is a target of many. These people assume this is now I showed you this already, but when I showed it in my lectures, I didn't turn it the full tour. So now I have to confess it because it will go. Pyroperoxide reductase, as I showed you, this was on the slide, has a number of functions. Proliferation, for example, very bad thing. Proliferation with formation of metastases, you know what I mean. Angiogenesis, we talk about what that is. But there are actually two subtypes of pyroperoxide reductase. Pyroperoxide 1 is the cyclical one, which is inside the cell. And then there is a second subtype, pyroperoxide reductase, which is specifically in the mitochondria. And this particular pyroperoxide reductase actually serves to inhibit apoptosis. So apoptosis, as we learned, is the natural way for the cells to divide. And if this is inhibited, it means that the cell is living forever. Cancer cells, potential. So this pyroperoxide reductase in the mitochondria actually makes cells live for very, very long. It makes tumor cells live very long and not die. So if we manage to inhibit this one, then we would get back apoptosis. Because the inhibition of apoptosis is like minus or minus plus apoptosis. And that's what we want. We want the cancer cells to grow up we want to go into mitochondria to get this target, but we don't want to do the other cell. So we need to be specific about mitochondria. This is an added layer of complexity compared to the core cell we have when we were talking about tumor cells or non-tumor cells. Now we have intracellular delivery to the mitochondria, not just any level, specifically mitochondria. How do we do that? There are mitochondria localizing types that were reported in the literature. For example, relatively simple combination of phenylalanine and arginine, so little bit positively charged amino acids. This is a minimal version that works for amino acid. This one here is slightly better. It works a little bit better. Now we need to put our metal complexes to one of these peptides. How do we do this? Just when we started this research, it was done by Professor and I introduced the degree. This paper here appeared in Organometallics and is good from training and co-workers. They show that this gold enzyme, the phosphine gold enzyme, reacts spontaneously without any further addition with an alkyne, an aromatic alkyne, forming this hydrogen, so-called triazole on the other end. You can see this is quite stable. It's an organically gold compound now. It's a carbon gold bond here, so it's an organically gold compound. It's quite a stable compound. And this, when we saw this before, this is fantastic. With this, we can get this done on peptides. We can introduce such an alkyne chemically into a peptide. Because this amino acid is known, this is phenylalanine. You can use phenylalanine with the which is para-actinated phenylalanine. Nowadays, I think it's actually commercial. At the time when we did this, we actually had to make this amino acid, but it's not a problem. It can be made protected by phenylalanine group, and it's incorporated in the peptide, and all the time peptide synthesis, and it's the same as the peptide. Even on the resin, you can deprotect the TMS group, so you get free alkyne, and the free alkyne reacts with the gold complex, and we're getting this triazole now on the leave this, and what can you say? This gold carbon bond is actually a very stable molecule. It's stable in acid, as you saw. It's stable in air. It's stable in water. It's a very robust compound. The thing we want to look at is this compound here, the phosphine, diethylphenylphosphine, which we term EK100. The second compound that Gabi Professor that we made. It's EK122. The first thing we did is we treated cells with this peptide called carbonate, and this is cells that are living under sort of controlled conditions, and we could measure the number of cellular parameters. For example, this standard cell adheres, this means how well the cell adhered to the surface, and we can also make oxygen consumption or respiration rate, so if they're breathing normally using oxygen, then everything is okay. What you can see here is that we treat with our compound, and then we have to look at 1K122, it's this blue line here, the red arrow. We look at this one here. Within 24 hours, the cells have 100% increase in healthy, and in 24 hours it goes down to almost zero, so they're all dying. Same for oxygen consumption. Here, at this time, they're not using any oxygen anymore, which means we are killing the compound here within a certain time. I should be honest and say also the cyclic phosphine gold chlorides do the same, but they do it in a much faster way. And basically, this is like an emergency, like a suicide, jumping on the bridge. So here the compound really shut off the cellular machinery very, very quickly. Whereas this one here, this 24 hours is the right time, is actually worked by a cell biologist, This looks like a regulated process. So things are initiated, they are processed, within a day or two or so, the cells will shut down in a regulated manner. Therefore, hopefully, we will demonstrate for this one. You can translate this curve here in a traditional so-called IC50 value to get quantitative healing. And so-called IC50, which means it's the concentration at which 50% of the cells are dying, is in the order of say 10 to 30 micromolar. What does that number mean? It sounds okay, it's actually kind of okay. Typically, if we use cisplatin on the cell line, this number would be between 2 and 10 microns. So cisplatin is more potent, it's lower concentration than 50% of the cells, but 10 to 30 is also not so bad. Keep the numbers in mind, we'll come back to that in just a second. So the key question is, does the gold compound actually do what we expect it to do? We studied pyridoxine-lactase inhibition in comparison also to glutathione, and we already know pyridoxine-lactase is selenium, here are the numbers. The affinity of our compound for glutathione-lactase is nanomolar, 20 nanomolar, very important. Glutathione-lactase is 460. So according to the first 20 times higher, more than 20 times higher. So clearly there is a high affinity for selenium. If we inhibit pyridoxine-lactase, it's a redox system, remember, then within a very short time, actually reactive oxygen species go up in concentration. With more compound, we inhibit more vinegar, we get more reactive oxygen species. Also, we change the mitochondrial membrane potential. So this is a clear indication that yes, we are actually working on the mitochondria. We are getting reactive oxygen species and we're getting apoptosis. We can prove this by measuring apoptosis. This is with an assay called NH2 staining, iodide staining, and we have to look at the white bar. The bigger the white bar, the more apoptosis we get. Already see at 20 to 40 micromolar, we get up to 80% apoptotic cells. So we're getting exactly what we are expecting. We're working on the mitochondria, we are inhibiting the enzyme, we're getting reactive oxygen species, and we drive the cells into apoptosis, exactly what we want. It's stopping the inhibition. We can actually prove that we are getting into the mitochondria by using the slightly longer peptide, where we also put on the fluorescent light. Here you can see again the phenol element, the triple bond, the gold on it. So this is our gold functionalized peptide, slightly longer now with the fluorescent light on it. And you can really see really nicely mitochondrial uptake. This is our compound again. This is the mitotracker line, which specifically localizes, and if you overlay, you see almost 100% gene expression in yellow. So this is the merge picture, you see only yellow. So we have almost perfect localization in the mitochondria. So this was a really nice result. Now, think back to the lectures that we heard. We are talking about a mechanism that involves mitochondrial localization and mitochondrial initiation of the full mitochondria. Compare this to cisplatin. Cisplatin, we know, works in the DNA. It inhibits transcription of DNA and so on and so on. So, if we really have a different mechanism of action, then we should be able to use our whole complex and we should still be able to kill cells which are resistant to cisplatin. If they have developed some mechanism to stop this DNA repair and the DNA thing from being from cisplatin, they should still be susceptible to our immune mechanism talking about mitochondria. Okay? And here's the proof of the concept. This is now a cell line, the P53 cell line called MEA-ME231, and I think the cell line also came out from Gerard Aulet's compound. This is one of the really aggressive breast cancer cell line that does not listen to the receptor, and it also is resistant to cisplatin. You can see this structural line here is treatment of the cell with 20 micromolar cisplatin. You remember the numbers I just gave you? I said in these normal cell lines, 2 to 10 micromolar would be an isotonic value for cisplatin for the cells. Now, this one we're using 20, so like 10 times as much, and the cells still will have resistance to it. Cisplatin is ineffective against the cell line. But we give our compound red line. Even in this cell line, it's very aggressive complex. We see this move cell respiration. That is respiration or acidification or it's the same picture. Within 24 hours here, this seems to go up first, this is like an emergency reaction, but then after 24 hours, so You can actually kill cells that are resistant to cisplatin, which is very useful. Resistance to cisplatin is a major problem. And we can understand what we do this because we're attacking it with the time. We're bypassing the resistance mechanism that exists in the system. And we are doing this by directing our compound away from the DNA, directly into the mitochondria, where we expect a different So I guess this might answer some of the questions you have. How can we do this? It's just an example. I think of many other examples. You can probably come up with smart ideas of what to do. But this is one example where we have used this. And we have, as you can see, the science to bring in a lot of cell biology that we will develop to demonstrate from the beginning to the end everything is there, as you can see. We have 5 more minutes. Yeah. And it should be done. I want to give one last example of how we can target tumors in a slightly different way from what we've seen so far. And this is a family of peptides that were developed by Roger Sand. This is the man who developed these things. Roger won the Nobel Prize. In fact, a lot of these concepts like for the development of reciprocal interpretation of reciprocity, he actually is easy. Roger, about 15 years ago, developed a very clever concept. How can we get things specifically in tumor tissue? We wanted to activate tumor-active compounds, tumor-active peptides, selectively in tumor tissue. And I'm stressing again the tissue, not the cell. As you will see, remember I talked about pH, tumor tissue as well. This here is a similar thing. So what we have is we have a payload, say, toxic compound, for example, attached to a polycationic peptide, a polyarginine. These polyarginine sequences are known to be taken up extremely well. These are some of these cell-penetrating peptides that are very rapidly incorporated into cells. Unfortunately, they're not specific. They're taken up by any cell. Okay, so that's all we want. We want tumor specificity of any cell. Therefore, the polycation part, polyarginine, is masked by the polyglutamic acid, which is anion, it's the same number of characters. So in total, this conjugate here is neutral, and it's of course because it's very large, it's not taken up by cells. But then, if this lower part can be cleaved by proteases, and if this happens in the tumor tissue, then selectively where the tumor is, we remove the anionic shielding part and we get the polycationic peptide, the payload back, and this is taken up, and then because we're now in tumor tissue, then it diffuses into the cells like magic. So the trick is that it is known that tumor tissue expresses so-called metallomatrix proteases. They are overexpressed in many tumor tissues. What they do is they are matrix proteases, as the name isolate, which have the surrounding matrix around the tumor tissue that is there, because the tumor cells need to break neurons to grow. And they do this by treating the proteases which chew up the exterior, and then the tumor cells go more rapidly. So that's what these NFP2 and NFP2 in particular has been linked to some very bad tumors like poor prognosis and breast cancer. So if we have a peptide sequence here that is recognized by NFP2, we can have something that's selected for breast cancer. That's the addition to this. Again, activate outside the cell, and then only a part that one that is activated is incorporated. Here's again the picture of what we did. We used again gold compounds because we knew they were active. In this case, we used cyclomethyl-3 compounds. We also see some of those on the slides. We know they are active on other literature, so we just look what is in the literature. We leave them in the peptide. We have a small peptide binding polyarginine sequence, and we have the full-length peptide with the NFP sequence and the polyglutamic acid. And they all swim around outside the cell. This is NFP2. NFP2 attacks and shoots up here the unique sequence, and then we get this sequence up, and only this sequence is taken up into the cells, and then we get the toxic effect from this. just to give you a glimpse, this is the kind of analytical work that we do. So this peptide synthesis was successful. The CAH will see one single peak after purification, so it's a very pure peptide. It has some fragmentation patterns. We can assign all these. We use some ligands. This is the full length peptide. It's coordinated. So with the mass spectrometry, it's not very pretty mass spectrum, but it's relatively neat, especially HPLC that is pure. Okay. First of all, we need to establish cell lines that we have MMP excretion. And we already know this. We did the quantitative PCR as we did for the receptor. You see here relatively high amounts of the messenger RNA PCR. For example, the HeLa cells, in these breast cancer cells, and then again NDA and B468, similar to NDA and B431, which we previously is one of the bad guys that has very little of RNA. And it's not just the MMP is there, it also needs to be active, and we determine this by these color colorimetric method where you can see we have this is the NDA and the one breast cancer cell line. You can see there's actually very little of the active protein. There's the other one, HeLa and ST, T, that's this one, actually a lot of it. So it looks like we're ready to go. We did the experiment, we looked at the IC50 values, and this is a horrible table to look at, but basically it tells you that we didn't have a spectacular effect. So we had cisplatin in that only between 2 and 10 or 20 maybe. You can see here, this is a bad cell line, has an IC50 value of about 20 as compared to HeLa and this one which has much lower. See, this is more resistant. And if you look at the gold compound to start with, it isn't very active. And also the peptide is anti-arginine sequence, the polypeptide, the one that should be taken up easily. We can see here, yeah, okay, there is some activity, not as good as cisplatin, but again, the resistant peptide is much higher as a peptide. And then if we mask this peptide, you can see in this one, the H570T, the activity actually goes up significantly, which is kind of bad news because this should be activated, this should be free, and this should be activated with this one on the cell line. And here, you don't see an effect, that's kind of expected as well, because here the cell line doesn't actually happen. So this is kind of inconclusive. This is not really what we expected. We were kind of disappointed when we saw this term. So yeah, we had some activity, but not exactly in the way that we really expected this. So we thought of something else. And how can we possibly improve? I mean, there are many things that people wrong with, right? So maybe we didn't play long enough, maybe he wasn't quite active, and so on and so on. What is an experiment that we could carry out to get further insight? And what we did is we added an inhibitor of MMP2 activity. So what do you think would happen if we add to all these different cell lines, the HeLa, the HS, and the NDA, and so on, so how would we do if we add inhibition? Well, the MMP2 cell line, of course, if we inhibit the enzyme, then we would get no psychosis feedback. Whereas for the other cell line, it would actually make a big difference. And here are the data that we get. The HeLa cell line is the one that has the overexpressed. So you can see here, look at the carefully scientific. You have to look at it scientifically. So this is the 3 is the one with full length peptide, and 2 is the one that just has the polyarginine. So you need just to compare those two. Polyarginine shouldn't make a difference because we don't need the activity here. That should be the same. Whereas in the full length peptide, we should see a difference because here MMP is there, and if the inhibitor is there, then nothing happens. So if you look at this, we are adding the polyarginine and the full length. So both are active to some extent. There is no effect on the polyarginine sequence if we add the inhibitor. Here the effect. But if we add the inhibitor to the length, then suddenly the population is not is not active anymore because we are actually inhibiting this enzyme. So you really have to look at this difference here. So you inhibited enzyme, activity, and the non-inhibited actually is not working. So I think this is a pretty elegant experiment which actually shows that yes, we are actually addressing NMP2 when we talk about the reactivity of this one. Admittedly, the reactivity is not very high, so we are not yet at the active compound, but we certainly have a distinct effect here of NMP2. Now look at the NDA and B cell line, one that has a lot less of the NMP2, and exactly that is what we see. So here the difference between the inhibited NMP2 and the non-inhibited is maybe just a factor of 2 or so maximum. So there isn't actually much of a difference. It's overall less active, fine, when you use it anyway, which is cell line control, but also NMP inhibition will be very important. So to me, this is work in progress. We're still working on this. We're trying to find more active compounds to actually get the lower bar here, like this, really low high activity, so that we see more drastic difference. So this is really work in progress that I'm showing you. It's not yet published also. This will give you a glimpse of the kind of experiments that one can do to establish things like activity, selectivity, or tumor, or tissue, or uptake, and efficiency. I would like to briefly summarize what we have seen, what I showed you in this last lecture. We talked about the synthesis of a number of different conjugates, bisolid synthesis. I hope I showed you at least some examples that we are trying to do comprehensive characterization of the materials that we have, which is very important because of the chemistry, how of the purity and the identity of our compounds. We're not just mixing anything, but we really want to establish what compounds we have, what is the structure, what are the targets, and so on and so on. So we do this in all cases, and so we use a few examples. We've seen some new chemistry, the skin chemistry, the soil enzymes, for example, some intelligent chemistry, so things that we do typically in our lab. We have also done biological experiments, cellular uptake, live cell fluorescence microscopy, a lot of in-depth investigation of both compounds. This was actually collaboration with a colleague in our room, and then we worked with 200 of both compounds together in Chaperone, which actually on our campus to help us develop all these things. The work was done by co-workers in my group. I think I pointed out now Metzler. Sandra was involved in the two work that was published yet. These were the more actually Einstein and should also be an Indian co-worker who was on here who actually was Sandra or Axel. There was some Dutch people here in my group, and the Dutch people was using sort of part of the discussion. I pointed out Stefan Löwen, who was a co-worker who worked on the old project, and then Stefan Heinz, who worked on the old university, the last project. We get quite a lot of support, and typically when we have international postdocs, like for example, Nolte, who come here, then we get support from the Alexander Nils Foundation, which was something about with that, I'm at the end of my scientific lecture. Some of you may have asked, and some actually asked me this, where is actually Bochum located in Germany? Well, I'd like to show you. This is a map of Europe. This is England, so I actually don't have to look more. So this part is not Europe, we decided to don't do it anymore, so just remove this. It's still comfortable for that. Okay. This is mainland Europe. France is down here. Paris is in the middle of France. Paris is in the middle of France. This is Italy down here, which is for the directions, so they don't know what they want. Germany is in the middle of Europe. Munich is down here. Frankfurt is pretty much in the middle of the middle. Berlin, our capital, is actually far to the east. It's almost Poland already. And then our city, Bochum, is far to the left. Here's the Dutch border. We are, where the red arrow is 15 minutes away from Bonn. This is the Nord, Soccer, Germany. You might know, we probably in Dortmund, they lost yesterday in Salzburg. Salzburg is Austria, it's not even on the map. It's really embarrassing. I was part of this. Okay, right. We are close to the Dutch border, we are close to Düsseldorf Airport, 2 hours away from Frankfurt Airport, so really easy to reach, and we have to live with the This is a picture of the campus of Old University. As you can see, it's a large campus. We have about 40,000 students on this campus here. These are the research buildings. Here is one of the new ones. You can see it's the original layout. This is the central lecture hall here, the only ground building. This one is the council building. The nice thing is this sort of goes downhill and there's a lake down here. It's all very green. It's rains a lot in Germany. That's why it's so green. Let's go to the next slide here. There's a lake here. You can go rowing, you can go sailing. There's a golf course over to the right. There's a gate. If you look down from this is the central entrance of the university, the central bridge. This is a view up from the hill. There are some farmhouses, and this is the iconic view of our university. This was erected in 1965. Just after the war, it was the first new university in Germany after the war. Many other followed afterwards, but this was the first one. And much like this university, which was built directly after independence. This was not directly after the war, and that was well. They decided to need more educated people, and they built this university. With that, I would like to thank, and also we found this already, part-time working for all the Tract, so our colleagues get this encouraged, this is what we like, this is broaden and click on my office and request work. Click on my office, you can request that the paper be sent to my office. It doesn't always work, sometimes sometimes the central office deals with it, but it is the church who I'm guaranteeing you to accept the paper, but of course, at least you can guarantee to get it treated 100%. There's a scientist who knows how to work. Thanks very much for all your attention. Thank you so much for all the questions. I really appreciate it. And I'm still here to it, so a lot of questions and things like that. Thanks again for attending this course. Also, please have a large attendance, even to the last day, Friday afternoon at 6. If I go back to my university, see how many students are still there, listening to lectures. I think we can have questions. Yeah, very good. Take questions and comments.
Sir, you're talking about the board peptide committee that acted on the anti-MD51.
Yeah. Yeah.
Sir, does this act by activation or anyone to wipe it from the PPT?
Well, the question was about the relation of the peptide and P53. The induction of apoptosis is a downstream effect, and I don't think that all peptides are working directly on P53. What they are doing is they are changing the mitochondrial potential, and then cytochrome C is released as a consequence, and so on, and then P53 is upregulated as a consequence. But all peptides are, I think, check this, but I think they are working directly on P53. They're working on the mitochondrial membrane. Then it is happening outside. You check for that, it is actually upregulated, but I think it's a leader. Yes. So the question was about this respiration rate measurements and circulation measurements. So it is indeed a continuous flow from the cell. So the cell is connected to a little vial where all the nutrients come out, and the compound as well, and constant concentration, and it's not the medium is constantly pumped through the cells or over the cells or not. The good thing is that we can measure real time the effect on the cells. When we do an IC50 assay, like cytotox assay, it's always an endpoint. So looking after 48 hours, 2 hours, we never see what's going on in between. you can see that with the growth hormone, different growth hormones, it actually made a difference. Some are actually killing the cells within literally 1 hour. And the radio compound, 1 hour later, all cells are dead. And to the cell biologist, that was needed. That just means that you are shutting off vital enzymes, and then the cell basically goes dead right away. Whereas what we were doing is really, it takes a day or so, it's really a regulated process. Question must until it gets upregulated, it takes some time. Maybe 2, 3, 4 months, 6 hours. And then you see an effect on it. So that's exactly what you see. You look on after CT. But the compound is active. And it's doing something, it's regulating something, and then it's molecule is. So this kind of experiment is very valuable to people who know how to look at these drugs. And for me, that's why I put the SPPU. But the cell biologists look at this and they are very encouraged. We are doing something very controlled. No. The cells are actually going confluently on the sensor, and the sensor has an oxygen sensor, a pH sensor, and it measures the impedance between 2 points. So this impedance is like cell adherence. When they're dead, they go up, and then of course, it goes down. If you can measure directly the pH in the medium, of course, it is very good. And you can also measure the oxygen saturation. It's like the use of oxygen. But this is done by separate sensors because you're a closed system. You can measure those numbers.
Okay, if there are no more questions, I would like to mention here that we Indians do believe in Kerala. Right? So we were lucky that you could be here for the last full week.
Hey, that was by intention.
No, but you were lucky to get hold of you guys, you pursued, and you could make it here and talk to us of medicinal and inorganic chemistry, and especially your last lecture, evidence that there inorganic chemistry or medicinal inorganic or bioinorganic chemistry is very useful. Taking you to peptides, organometallic compounds, which were originally thought to be untouchable, are now frequently used in such kind of studies for selectivity, for uptake, increasing the uptake in cells. This was never heard of before, or even not thought of, never dreamt of before, that metal complexes will be used for such kind of studies, research, and also for the application. I would like to thank on behalf of all of you, and thank Dr. Nils for coming here, spending some time here, and hopefully the next 2 3 days that you will be here, there will be more interactions. You are welcome to interact with him, discuss with him, talk to him, and also we have the lecture on Monday. And if you have any questions, you can still come. Still in the office number 305, and we can see you. Thank you very much. One o'clock.